Really is a pleasure to be part of this opening ceremony for GIA. It's been being planned for a bit, and it's nice to see that it officially launches. And I think it's going to benefit the community quite a bit. So my lab is primarily a biochemistry lab, and we've been focusing on glycosyl transferases for a long time. And the story that I'm going to tell you today is kind of the culmination of a lot of different aspects that I won't have time to give you the background for, but basically we've been concentrating on this idea that we could use a microbial factory to produce a human therapeutic protein with authentic human glycosylation if you manipulate enough genes. There we go. So one of the things that's really important to do is to thank your collaborators. And I wanted to point out a very long-term collaboration with Dr. Steve Withers at the University of British Columbia. We've actually been working together since I was a graduate student, and Steve is a great person for generating all kinds of ideas, and we've been working for quite a long time on this particular project. Number of trainees that have been involved. When I was in Toronto, I was at what was then Ryerson University and is now Toronto Metropolitan University, and a couple of the people from my lab there ventured out here to the Wild West. One of them is Nicole Thompson, and her work figures prominently in the story that I'm going to tell. And I'm going to briefly mention a collaboration that we have with Deanna Santer, who was here as a research associate in the lab of Michael Houghton in the Faculty of Medicine and Dentistry, and we have a collaboration looking at antiviral proteins that I'll touch on. So we were fortunate that Hans Wandel introduced the idea of O-Glycans earlier. But I wanted to reiterate that what we're interested in is those glycans that are prominent on proteins that have a therapeutic application. And what Professor Wandel told us about this morning was that they identified 15,000 sites where O-glycosylation occurs. That's a lot of sites. That's on a lot of proteins. But I want to narrow it down to those structures that have been seen on proteins that are either made recombinantly to use in a therapeutic space or are isolated from blood, for example, and used in therapy. And so we end up with a series of structures starting with that very simple, just a GalNAc on a serine or threonine, which is elongated to what we call a core 1 structure. And core 1 just means that there's beta-3 galactose on there. This is also sometimes called the T antigen. And then this, of course, can have my favorite sugar, sialic acid, added to it, not only in one place, but two, which makes it doubly exciting. And then you can start branching this GalNAc with some other sugars to make what's called a core 2 structure. And core 1 and core 2 structures are pretty much the most common so-called mucin-like structures that you would see on a therapeutic protein. There are a couple of instances where you see extended core 3 structures. And here are some examples of some further extensions that you can get. And again, Dr. Wandel showed some really complicated O-glycan structures. And we're not going to go there today, but there is some biology that happens with these simple structures. So we just put together, Nicole and I just had a review accepted on looking at the literature around glycans in therapeutic proteins, specifically the O-glycans. And this idea of what is the function of a glycan on a particular protein is something that a lot of people are working on, and it's difficult. And here's a very interesting example. So these coagulation factors, of course, are important for people with hemophilia. And there's a series of protease cascades that has to happen in order to get to blood clotting. And in that process, a number of enzymes complex, they have to activate each other, and they have to go on to do a variety of things. In the case where there's a protease called factor 10, which is activated into factor 10A, which then goes downstream to interact with thrombin, which then completes the cascade. There is this very short peptide in factor 10, which is called the activation peptide, and it contains 4 glycosylation sites. And nobody cares about the N-glycans. But we care about the O-Glycans. Actually, that's not true. One of these N-glycans does play a role, but I'm not going to tell you about it because I only care about the O-Glycans. So there are 2 O-Glycans here, and it turns out that in the complex of, there are 3 proteins that come together in order to activate factor 10. One of these O-Glycans is a specificity determinant. If this O-Glycan is missing, factor 10 is non-specifically converted to factor 10A. And that's a bad thing because you don't want that clotting cascade to happen independent of some event that would trigger the normal cascade. And so it's interesting that there must be then a protein-glycan interaction in that complex, which is responsible for, okay, I am the right protease to activate this particular protein at this particular time. And that is this little short O-Glycan. There's specific engagement in this complex of factor 8 and factor 9, which is involved in activating factor 10. There's a little bit of controversy of whether both of these sites are important, but literature seems to suggest that the O-Glycan at amino acid 17 is the critical one for the specificity. There is this really interesting protein that I didn't know a whole lot about until we started looking into this review article, and it's called the von Willebrand factor. And the von Willebrand factor is probably the most common protein involved in blood disorders. It's estimated that about 1% of the people in the US have some kind of problem with their von Willebrand factor. It's a gigantic protein. It has this really complex domain structure, and it polymerizes. It makes fibrils. And it's involved in homeostasis. And there's a particular domain, the A1 domain, which is down here, which is where the platelet interaction happens. And look what's down here surrounding the A1 domain. A cluster of serines and threonines. I wonder what glycan might go on there. So this cluster, and there was a really nice paper actually this year, which showed some really nice biophysical studies where this cluster of O-Glycans at the N-terminal side of this A1 domain stabilizes the domain. And it actually inhibits this platelet protein, glycoprotein 1b, from actually binding to this particular von Willebrand factor. It also turns out that the glycans at threonine 1255 and 1256, which are again here, are actually involved in plasma maintenance, which to me was, I think that's the first example that I could find of a protein in the literature for which an O-Glycan is linked to keeping a protein in solution in your blood to do its job. There's specific interaction with the glycan at serine 1486, again for platelet binding. And this plasma maintenance is actually a macrophage receptor, which kind of blew my mind because I tend to think of the liver receptor, hepatic receptor for clearance, but it turns out macrophages have this really nice protein called MGO. Many, many cytokines carry O-Glycans, and as Dr. Wandel pointed out this morning, there are a lot of proteins that have single sites for O-Glycans, and the cytokines, in some cases, have one site. Some cases, they have a little cluster. But when I started digging into the literature about what O-Glycans do for cytokines, things got really murky really fast. So for the graduate students in the office, in the room here, when you're citing a paper, make sure you go back and look at the primary literature, because there are papers that say that the O-Glycan on interferon alpha-2A protects it from proteolysis. And I defy you to find the primary data that says that. It's been propagated in a number of papers. I don't think that's true. So for those proteins for which the O-glycan has had a function ascribed, interleukin-2 was one of the very first therapeutic proteins that was made recombinantly. And people said, oh, it works without the glycan, we don't need it. But it turns out that what happens in IL-2 is that the glycan, the single O-glycan, right at the N-terminus of this protein, prevents it from aggregating. And when this protein aggregates, it becomes a very good antigen. And the people who were receiving it generate antibodies. And all of a sudden, this protein stops working because there's a good immune response to it. The similar kind of effect is seen in the cytokine GCSF, which is granulocyte colony stimulating factor. Again, single glycan. And again, the idea here is that there's a loop rigidification on this unstructured loop, which prevents this protein from unfolding, which would then lead to a polymeric aggregate, which is inactive. And there's some data there. There's a couple of papers which also suggest that it's responsible for protection from proteolysis, but again, it's a little bit murky. What do the O-glycans do on GCM, CSF, and interferon alpha 2b? Not known. I couldn't find any solid evidence for what they do. And a lot of people ignore them because these recombinant proteins, the ones that are in clinic, are made in E. coli and covered in polyethylene glycol. Right, so it's hard to know what the function is. So a quick summary then of a couple of engineered therapeutic proteins that are really kind of interesting. So it turns out that you can get O-glycans on recombinant therapeutic proteins. And these are proteins that are prepared in mammalian tissue culture. And there's this really interesting protein called the etanercept. And this is an engineered soluble TNF-alpha receptor. So it's an alternative way of mopping up TNF-alpha, which is, of course, an inflammatory cytokine. And it turns out that in this particular case, you get mono and disylated core 1, which I showed you. But you can also get this silated extended core 3 at several positions. So there's a lot of O-glycans on this protein. And it turns out that one of the effects here is an increased affinity for TNF-alpha. And that's kind of an interesting effect because why would it do that? I don't think we understand the mechanism by which the affinity for TNF-alpha is increased. Increase in serum half-life is well documented on this particular protein for these particular glycans. There's some interesting work that we ran into, which actually is quite old now. The originating work was quite old. Nicole was able to find some earlier work where people had taken the C-terminal peptide of the chorionic gonadotropin B protein, which is quite heavily O-glycosylated, and they used it as a fusion protein on a variety of things. And what you end up with in this particular case is the silated core 1, but basically on a peptide tag. So now you've tagged your protein with this site, and this leads to a nice increase in serum half-life. And actually one of these proteins that just got approved by Health Canada, so the human growth hormone with this tag, just approved by Health Canada. So again, engineering, looking for a simple silated core 1 glycan. And again, a similar effect for a sequence tagged interferon alpha 2b, although in that particular paper, we don't know which glycan was on it. Okay, so I think we're all familiar with this idea that glycans are important for proteins. And just a reminder that the typical thought is that this thing called the acyl-glycoprotein receptor is the hepatic lectin that removes proteins that have lost their sialic acid from the blood. And so then for a therapeutic protein, if you wanted to keep it in circulation, if you're capping with sialic acid, it should keep it out of the grips of the acyl-glycoprotein receptor. I wasn't really able to find too much evidence that O-glycans actually interact with this receptor. It's mostly N-glycans. However, there is this macrophage galactose lectin, which has been well documented to interact with O-Glycans. So then sialic acid is really an important terminal sugar on these therapeutic proteins to help with serum half-life. So as I mentioned, we're mostly a biochemistry group, and we've been playing with a bunch of different recombinant enzymes doing all kinds of in vitro glycosylations for quite a long time. But I realized that this idea of doing everything in a test tube probably wasn't going to fly, and that we should take advantage of the fact that a living organism would synthesize all of the precursors that you need, and you wouldn't have to buy them from Sigma and go broke. So we embarked on a program to engineer E. coli to take on the job of synthesizing the biochemical pathway to make sialylated core 1 structures. And this is a schematic of how that works. So most strains of E. coli don't make sialic acid. There are a few that make sialic acid, but we had to integrate this into the strain that we wanted to use. So this got inserted into the genome, and what this does is it takes UDP-glucnac, which is the precursor sugar that's really needed for pretty much every bacterium because it's needed for peptidoglycan. So we knew that there would be a good supply of this. This, in terms of helping with the O-Glycans, can be epimerized into UDP-GalNAc, which is then a substrate for the GalNAc transferase that actually puts that GalNAc onto the protein. And here's where we deviated a little bit from the script, where we're using a bacterial enzyme that normally works on a glycolipid to put this galactose onto the GalNAc to make the T antigen structure. And then we get back to the sialotransferases. And I'll tell you right now that the bacterial sialotransferases don't work in this case, but the human ones work really well. And that was a bit of a shock to us, but whatever, I'll take it, it works. Yeah, so this is the schematic that we had. How well does it work? So what we've done is we've taken a fusion protein approach. So our tester bed for therapeutic protein is interferon alpha 2b. And it's a good example because it's got 4 cysteines which form 2 disulfides, one of which is essential for the active version of the protein. But E. coli doesn't normally make disulfides in the cytoplasm. It makes them in the periplasm. So if you want to make them, if you want to make a human protein in the cytoplasm of E. coli, you need to use a special strain, and you have to play around with it a bit, and I'll explain how we approach that. We're also using a fusion to the B1 domain of protein G. And those of you who are in immunology will know protein G because it binds antibodies. This isn't involved in the antibody binding as far as I know, but it's a very small, stable entity. And it was actually somebody who works on protein NMR, Steve Smith at Queen's University, who introduced me to this because the NMR community has been using it as a fusion for human peptides in order to get enough protein to actually stick in an NMR tube. And we thought, well, okay, if it works for them to generate proteins, we should be able to do it too. And so this particular strategy works quite nicely. And this is a pseudo crystal structure. This is a Fire2 model of what that would look like. So there's a nice crystal structure for both of these, and then the model sort of gives you an idea of how this might look. And the glycan is up here. So easily accessible to the enzymes that need to do their job. So our 2 testbed proteins, as I mentioned, interferon alpha 2b, also human growth hormone. They both have similar folds, so they have these alpha helical bundles which characterize the basic fold. And this is common to most of the cytokines. And they both have an unstructured loop. And in the case of interferon alpha 2b, the natural glycosylation site is here. HGH also has this unstructured loop, but it's not glycosylated naturally at this site. So this is where protein engineering starts to come in. The idea being, we know that from the crystal structure of this sitting on its receptor, that this loop is not engaging the receptor at all. So we thought, well, we could stick a glycan on here and see if we can affect the properties of this particular protein. So what we started doing then was this idea of engineering a sequon, and many people are familiar with, N-link glycans have a sequon, so asparagine, any amino acid except for proline, threonine, serine, so NXTS. There's no such thing for an O-Glycan, but there are a few rules, and I'm sure Hans has seen this in many of the proteins. There's a lot of variation for where O-Glycans occur, but in our case, for these proteins, there's got to be a proline relatively close. No basic amino acids, although there's some exceptions to that. And aromatics don't help in our case. So what did we do? Well, we took the natural protein interferon alpha-2b. This is the glycosylation site. It's this threonine. And there's a very nice tool which was developed by Thomas Gerken at the University of Texas at El Paso called IsoGlyP. And IsoGlyP is a predictive tool that looks at the amino acid sequence and makes a prediction about the probability that a particular isozyme in the GalNAc-T family, how well that would work on a particular site. And interferon alpha-2b is predicted to work best with pp-GalNAc-T1, and we showed experimentally that that was true. The T2 score is pretty weak. Does it work? It works a little bit in our experimental system, but not great. So what we did was we played around with this sequence. We swapped the VGV for a PQP, changed the T2 score from 6 to 23, and now all of a sudden we get almost 100% glycosylation at that site. And that's a change from about 40% glycosylation with this score. So that was really nice. Now what do you do with a protein that has a score essentially of 0? We played around with it a little bit more. We ended up with this particular PT, and now we've got two potential sites of glycosylation with 2 scores, and go figure it's only the low score one that's modified. So we have a lot to learn, but it's still a good predictive tool. Okay, so then we embarked on a program to put together these synthetic operons that carry all the genes that are required to make a particular glycan. So we had, in its simplest case, the GalNAc-T and the Gal-T and a folding chaperone, either the E. coli one or the human protein disulfide isomerase. And then we expanded that to include silotransferase in different positions, so either the porcine ST3-Gal1 that we worked on, or bacterial one, CSD1, which is in completely different silotransferase family, and just sort of different variations to try to make this silated T antigen structure. And the only one that works is this combination of the human GalNAc-T2, the porcine silotransferase, and oddly enough, the bacterial Gal-T. We did try to put a cosmic independent Drosophila core 1 synthase in, but it doesn't work in E. coli. And we think that's because there's limiting manganese, and it's a manganese-dependent enzyme. And so there's a trace of activity. It makes perfectly functional protein. You can purify the protein, and it works great in a test tube. It just doesn't work in the operon scenario. We then went on to add enzymes to make the disilo structure. So either SD6-GalNAc-4 or SD6-GalNAc-2, again, both from the human case. And these work equally well, although SD6-GalNAc-4 seems to work slightly better. So now what we have is an E. coli strain carrying these genes on a plasmid, the sialic acid synthesis genes in the genome, and a second plasmid with the target protein on it. Just to remind you, what we're doing is we're taking the yellow box, we're adding a yellow circle, and then we're putting a purple diamond on, and in some cases, two purple diamonds. So it's a relatively simple scheme, but it involves a bunch of different enzymes. So what happens when you do this? Well, this is a summary of an experiment where we actually grew up 1 liter cultures of these E. coli strains. So we're working with about 10 grams of cell mass. We were able to isolate somewhere in the neighborhood of 40 to 50 milligrams of the protein. And then the question was, well, how much of it's modified? If we do small scale cultures, we can get 100% modification. So a 50 mil culture gives you really good glycosylation, but of course that's not gonna work for therapeutic production. You're gonna have to go to a fermenter probably. So this we thought would be a good test of the robustness of the system. This is an HPLC analysis that Nicole developed in my lab using a reverse phase setup where the unmodified protein is the most hydrophobic, and as you add glycans, it comes out earlier and earlier. And the first thing that you'll see from these HPLC traces is that the amount of unmodified protein, so no GalNAc, is really a problem. But there's not a lot of intermediates. So we don't see any T antigen intermediate when the silotransferases are there. So these silotransferases are hanging out, and as soon as there's a T antigen, they modify it. So they're happy and ready to go. We get a little bit of just the GalNAc, but it's this proportion of the unmodified protein that we're a bit concerned about, and it's just quantitated here. So in the best case scenario, we can get somewhere in the order of 60% of the glycan to be the disilo T antigen, and this is on the seq1 modified protein. And it's not quite as good for HGH, but remember, that's a totally synthetic seq1. All right, so once you've sort of gotten addicted to playing around with seq1s, you start going crazy. And so we made a whole bunch of different loop mutations for HGH, which isn't modified as well, with a variety of different scores, some of them really high, again, some of them mixed. And so this was our first foray, remember, and we found out that this low score is the major modification. So eventually we ended up with one that just has a single threonine, because we're getting smarter as we go along. This has got a pretty good score. Okay, so does it work, and what happens? So remember that the mutation number 1 to give us this gave us around 50% unmodified protein. And what Nicole was able to show was that when we looked at version 2 and version 3, and now just starting off with either the human GalNAc-T or the Drosophila GalNAc-T ortholog of that, or the Caenorhabdites ortholog of the human protein. And in a combined reaction, so this is now actually a combined reaction, you can actually get really good modification relative to mutation number 1, surprisingly with the Drosophila enzyme. So we've created a really nice seq1 for the Drosophila enzyme, but it's still very good for the human one, certainly much better than the original modification that we made. So we're still playing around with these. The reason I didn't show you the HPLC for this is because this HPLC method is kind of sensitive, and these mutants actually don't resolve very well. So we just get a blobogram, which isn't informative. You have to really squint at it and turn your head sideways to make heads or tails out of it. Okay, so what are we doing next? We want to look at different core types because these have potential for longer chains. We want to look at this idea of multiple sites. So seq1 tag, I know it's been done before, but not in the E. coli context. And the idea I'm going to talk a little bit about, we've been thinking about how would you make a synthetic mucin type structure that has multiple glycosylations. And we're sort of halfway there. we want to look at, does this modification do anything to the half-life? We did a preliminary experiment with our colleagues at NRC in Ottawa, and it didn't look like we got much effect, but we need to repeat that experiment. We also need to look at this idea of prevention of aggregation. That's something. And eventually, we're going to get rid of this second plasmid and try to put as much stuff into the genome as we can. All right, so this idea of can we make multiple O-Glycans on a particular peptide, and I know some of the early work from Hans and other people at Copenhagen, a lot of these in vitro assays have been done, but we have a slightly different idea, I guess, behind what we wanted to do. So we started off with this very simple Muc1 repeat that has two adjacent residues, a serine with a terrible score and a threonine with a okay score. And in vitro, both of those sites are modified by GalNAc-T2, no problem. So we can get the di-GalNAc product. Then we can use, in this case, the Drosophila core-1 synthase to make di-T antigen. So we can go from 2 GalNAcs to 2 T antigens, and that's nice because they're on opposite sides of the peptide backbone. So we can actually do that. So we can go from, so we can put the T antigen on. What we're shooting for at the moment is core 2, because we have a collaborator who asked us, can you make this structure on the Muc1, because we think we have a protease that recognizes this structure. And the answer to that question is yes, we can do this. This is very cool. So this sort of leads into what other uses do these peptides have? And we've had a long-standing collaboration with Al Borston's lab at the University of Victoria, where Al is looking at a family of glycoproteases from bacterial pathogens. And we've been trying to map the specificity of those. And so we need a library of compounds. And so we're now working on Muc1 repeat, Muc2 repeat, Muc5 AC repeat, and making some nice progress on being able to provide him with substrates that have very defined glycan structures, so that in a sort of semi-high throughput fashion, he can look at the specificity of those particular proteins.